Hello, welcome to this lecture of Biomathematics. As we have been in the last few lectures, we have been discussing differentiation, and in the last lecture we discussed its application in biology. We discussed how certain filaments grow and the knowledge of derivative, how does it help us to understand about the rates, etc., etc. So, we for example, knowing the rate of growth of actin, we could get some idea about the filament growth and then the process of growth of filament. So, in this lecture we will continue learning some more applications of biology and how we can apply the idea of derivative to understand some more problems in biology. One more or two more examples we will take, and then we will go to the next topic. So, as you can see, today's lecture again is differentiation and its applications, and we will discuss example 2 in the application of biology. So, the example 2 is enthalpy and entropy of a chemical reaction. So, as you all know, one of the important thing in biology is chemical processes. So, any chemical reaction, understanding of chemical reactions and things related to that is very important in understanding various processes in biology. So, let us take here a simplest possible chemical reaction and let us try and understand a bit thermodynamics of it and how does this knowledge of mathematics will help us there. So, one example, one thing here is, so let us take the simplest case as you see in the slide here. Let us take the case A going to B and B goes to A back. So, this is A goes to B and back. So, this is a reaction A arrow forward arrow B and backward arrow this way. So, what does it mean is that, as you all know, A is converting to B and B is converting back to A. So, this A and B could be two states of a protein, proteins having state A and state B. It could be an enzyme changing something from A to B. It could be anything you can imagine. And if you have any such a reaction where A goes to B, then you all know that the equilibrium constant, the K equilibrium for such reaction, as you see here, is K equilibrium is equal to the concentration of B at equilibrium divided by the concentration of A at equilibrium. So, when I say equilibrium, let me briefly tell you, equilibrium means the rate of A changing to B is equal to the rate of B changing to A. That means, some amount of A is getting converted to B, some amount of B is getting converted to A, but the concentration of A and B on an average does not change. That is what we call equilibrium. So, at equilibrium, when the concentration of A and the concentration of B on an average do not change, the equilibrium constant is basically the ratio of the concentrations of B divided by the concentration of A. So, this bracket means, as you know, it is concentration. So, the concentration of B at equilibrium divided by the concentration of A at equilibrium, and this constant is known as equilibrium constant. So, now another thing which we all know from thermodynamics is that delta G. So, the delta G of a chemical reaction basically is the change in energy or the change in free energy actually for the chemical reaction. And we all know, you might have studied this, that the formula, the delta G 0 is basically minus R T log K equilibrium. This is the formula for delta G 0. Now, you also know that delta G 0 can be also written as delta H 0 minus T delta S. So, free energy is enthalpy minus temperature into entropy. This is also you know. So, if you combine this two, you can substitute instead of this delta G 0 here, you can substitute this minus R T log K equilibrium from the top equation. So, combining this equation and this equation, I substitute this here and I get R T log K equilibrium is delta H 0 minus T delta S. And if I rearrange this, that is, I take minus R T to the right hand side. So, I divided throughout by minus RT, I will get ln k equilibrium is equal to delta H 0 divided by minus RT and minus T delta S divided by minus RT. So, essentially you will get ln k equilibrium is equal to minus delta H 0 divided by RT plus delta S by R. So, these two equations combining, we can rewrite such an equation which is very simple, just simple algebra. One can rewrite an equation ln k equilibrium is equal to minus delta H 0 divided by RT plus delta S divided by R. Now, for a chemical reaction, if you think of, like typically the chemical reaction happens at a fixed temperature. So, in biology, like it is like 310 Kelvin or 37 degrees Celsius, the temperature is a constant. R is a universal constant. So, R is a constant, T is a constant. We can calculate ln k equilibrium. So, let us, for any reaction, you can calculate ln k equilibrium. Now, if we vary the temperature, so if you imagine an experiment in a laboratory, one can actually control the temperature. So, let us say you vary the temperature from 25 degree to 37 degrees, 25, 26, 27, 28. So, as you vary the temperature, you can imagine that ln k equilibrium also varies. So, as we vary the temperature, the equilibrium constant also varies. That means, ln k equilibrium changes. So, essentially one can imagine doing an experiment where you keep a temperature at 25 degrees Celsius and do the reaction A going to B, and when it reaches equilibrium, calculate the ratio of B and A concentrations and you will get ln k equilibrium. So, and write it down. So, let us think of a table. So, let us think of, so if you do this experiment, you can draw a table. So, temperature versus ln k equilibrium. So, let us say you do this temperature at, let us say 283 Kelvin. So, so 283 Kelvin, you do this and you start the experiment. A will go to B and back, and when it reaches equilibrium, you can calculate the concentration B by A and that value and log of this, and this is basically k equilibrium and the log of this is basically, you can write it here, log of k equilibrium. Now, let us say you do it 285 Kelvin, you calculate the ratio, again do the experiment, know the concentration at equilibrium of B and A, write down this number here. Like you can vary 287, 289, 291, so on and so forth. So, up to 310 you can just do and calculate this value. Then, once you get such a table, you can plot a graph. The graph is, so let us say you plot a graph which is 1 over temperature versus ln k equilibrium. So, let us see what does it graph give us. So, 1 over T. So, as we know the T here, as we know we did T is 283 is the first temperature we did. So, here you will have 1 over 283, here you will have 1 over 285. So, all these are numbers. So, you just for all this number you will have a corresponding k equilibrium value. So, for 1 over 283, 1 over 285, 1 over 287. So, as you vary 1 over T, you will have ln k equilibrium varying. So, let us see how does the ln k equilibrium vary. So, let us look at the next slide. So, the typical graph will look like this, it is like this blue line. So, for as the 1 over T increases, that is as the temperature decreases, the ln k equilibrium will decrease. So, if you look at this equation, so let us look at this equation. So, the equation we had was ln k equilibrium is equal to minus delta H 0 by R into 1 over T. So, as you can see here, delta H 0 by R T can be written as delta H 0 by R into 1 over T. So, let us write it delta H 0 by R into 1 over T plus delta S by R. So, this way you can rewrite this equation and then you can plot. So, now ln k equilibrium versus 1 over T. So, this if you look at this equation, this is the y. So, this equation looks like y is equal to some constant minus sign with some constant m, 1 over T is your x axis. So, this is x plus some other constant c. So, this is a straight line with a minus m. So, this is a straight line, as you all know, this is slope. So, this equation essentially gives a straight line with a negative slope. So, that is what you have here. So, if you look at this graph, straight line with a negative slope is essentially this. So, this graph as you see here, this graph is basically straight line with a negative slope. So, the derivative, in other words slope is nothing but, so we learn that slope is nothing but derivative. So, the derivative if we calculate is basically dy by dx. How does the y, that is ln k equilibrium, changes when things in the x axis changes. So, it turns out that if you calculate this dy by dx, as you can see from this equation, if you take this y is equal to mx plus c and calculate the slope, the derivative, you will see that the derivative is minus delta H 0 by R. In other words, the slope gives you enthalpy divided by R. So, if since we know the R is a number which we all know, we can easily calculate enthalpy. So, by calculating the derivative, we can calculate delta H 0. So, that is the message here. So, from this such an experiment, by calculating the derivative of this curve, we can calculate the enthalpy delta H 0. And as you can see here, the y intercept, that is the value of y at for this particular x axis, for the value of y when 1 over T is 0, basically is basically delta S by R. So, that will give you some idea of entropy also. So, you can, since this is a straight line, you can extend the straight line up to the value where 1 over T is 0, and at that point you will get the entropy delta S by R. So, from this particular experiment, by knowing the simple idea of that derivative is nothing but the slope, and by calculating the derivative of this curve, experimental curve, we can get the enthalpy and enthalpy delta H 0, and from the y intercept we can also calculate the entropy. So, enthalpy and entropy from the simple experiment where we vary the temperature and measure the concentration of A and B. So, if you just sit back and think, what did you do is nothing but vary the temperature and calculate the concentrations of A and B at equilibrium. From the simple experiment, and by plotting this and using this idea of derivative or slope in mathematics, we can get what is the enthalpy delta H 0. We can also calculate delta S. So, let us, so this is another example where the idea of mathematics, the idea of derivative, the mathematical idea of derivative is used in biology to get some information. Now, we will go to the another one more example. So, when we were discussing the idea of function, we were discussing many function, we actually discussed an example of population growth. So, we said an example of fish growth. So, the example was this, let me just remind you, the example was this, that let us say you are doing some farming, fish farming. So, you have a big pond and you add a lot of fish and the fish grows. So, it takes a few days for the fishes to grow. So, you get many fishes. So, the number, if you plot the number of fish versus the time or the density of fishes, how many fishes are there in a unit area or unit volume, that is, we can call it density, the population density of fish. So, if you plot the population density versus time, we had got the following graph. So, let us look at this graph which we had got a few lectures ago. When we were discussing functions, we had shown this graph, which is basically population density versus time. So, what does this mean? So, this means is that first when you grow the fish, first the time is, let us take this time in days. So, time is this x axis in days. So, first the fish population is very small and increasing very slowly, and the population density increases here and then it reaches a constant value. So, this is the typical thing which you see in any growth curve. And we wanted to understand this question, that at what population density we will get the maximum yield. So, what do you mean by maximum yield? When we say maximum yield, what we want? We want the number of fishes to increase very fast. So, the growth, the fish growth has to be fastest. So, that is where you will get the maximum fish. Like if you had like 10,000 fish in the whole pond today, tomorrow you want it to be 20,000, 30,000, the more the better. So, in one day, how many fish will it increase? So, the change in number of fish in one day, that is what yield, right? I mean, the increase in number of fish in one day, that is the yield you get. So, and so this idea of change in number of fish when you change the time is basically, you can saw this like derivative. So, let us think about this curve a bit more. So, the curve basically y is number of fish and this is time. Now, as you can see here, this has a shape which is something like this. Now, if you change this, let us say write the days here 0, 1, 2, 3, 4, 5, 6. So, these are days. So, in a week this gets saturated, let us say this is the case. So, now if you just look the first day, the 0, so this is 1, this is 2. So, if you just look here, the increase is actually very small. If you just wait this one day, the number increases only a little. On the other hand, if you look here, somewhere here, between second and third day, the increase was a lot. The number increased from second day, the number was this, the third day the number is this. So, the number increased a lot in one day here. So, the yield was maximum in this particular region. So, so if you call this change in time, so you call delta t, that is this delta t, and this is delta n, change in number, delta n by delta t is basically the yield. So, this is basically the derivative. In other words, when the delta t is very small, I can call it dn by dt, the yield. So, this is the dn by dt. So, the slope or the derivative gives you the yield. So, think about it once more. Yield means you want the maximum number, the delta n, the number should increase when the time changes by 1 day, by say time changes by a small factor. If you look here, if the time changes from by 1 day here, the yield was very small, the number only increased from here to here, the number only increased this much. On the other hand, from here to here, the number increased this much. So, as you can see here, as you can see in this graph here, if you keep calculating the slope from here to here, you will find that in the middle somewhere here the slope is maximum. Slope is maximum somewhere in the middle. So, that is the place where you get maximum yield. That is, in 1 day, in unit time, how much more fish you get, that is the yield. So, this idea of derivative essentially helped us to understand where do we get the maximum yield, like at what time. So, that at this particular time, after the here you can say after the second day, if you look at this, after the second day, if you get the maximum yield here in this graph. Here also somewhere here is you will get the maximum yield. So, if we could, by calculating the derivative of the curve at various times and looking at where the derivative is maximum, where the derivative is large, the bigger number, where the slope is maximum, you will get some idea about where the growth is maximum. And when the growth is maximum, when the number is, growth means the increase in number, or the number of fish or the number of any organism increases very fast, you can say the yield is maximum or the large yield. So, here again the idea of derivative helps us to understand something about growth processes. Now, talking about the growth process, one more thing I want to discuss today is that typically when you plot a growth curve, so the growth, when I say growth is very fast, typically what one means is that the growth is exponential. So, when you say fast, very fast growth, what does it mean? So, let us have a look at it. Let us just revisit this bacterial growth, what we had discussed, and then look at what does it mean fast growth. And you also might have used this log phase sometime, the word log phase. Why log? So, those kind of things we will briefly discuss in this context. And so, let us go back and revisit this bacterial growth a bit, because this is one important area in biology. We will also come back to this, because growth and decay processes is something which is very important thing in biology. We will discuss more and more about this, but for the moment let us since we now understand the idea of derivative, let us revisit this bacterial growth. So, let us go to look at this slide here. We had seen this slide. So, we are discussing about bacterial growth. Let us take the example of E. coli. So, to begin with, let us say you have one E. coli, and in about 20 minutes the E. coli will divide into two. So, you will have two E. coli. So, this is the first generation, one generation, the next. So, 20 minutes is over. So, it is 2 power 1. The number of bacteria is 2 power 1, which is 2. Now, in the second generation, which is this, another 20 minutes if you wait roughly, you will get 4, this will divide into 2, this will divide into 2. So, you have this 4. So, which is 2 power 2, the second generation. So, everything is divided into 2, that is this 2, and this power is for the generations 2 power 2, you have 4. In the third generation, you have 2 power 3 number of bacteria, that is this will divide into 2, this will divide into 2, this will divide into 2, this will divide into 2. So, you have total 8 bacteria, which is 2 power 8. So, 8 E. coli, which is 2 power 3 equals, which is 2 power 3 equal to 8. So, this is the way bacteria grow, 2 power something, because everything is dividing into 2. So, this is 2 power some factor. So, this we call it exponential growth, because 2 power 2 in the exponent. So, the number, the bacterial number increases as 2 power some factor. So, this is you exponentiate, like this is the power. So, this is anything in the power is called exponent. So, some exponent of 2 is the number. So, now as we had said this last time in the slide, number of bacteria at any time t is given by 2 power k t. So, here you have k t is the exponent, where k is the rate of cell division. For bacteria is typically 1 in 20 minutes. So, k is 1 over 20, t is the time. So, if you take t in time in minutes, k is 1 over 20 minutes. This is n of t is e power k t, which is t by 20. So, here you have t divided by 20. So, let me write it again more clearly. So, then at any time t is 2 power t divided by 20, where t is time in minutes. So, this is the kind of formula which this is the kind of function which we discussed last time, the number function for the bacterial growth. So, known the t, you can calculate this function. So, for example, let us take t is equal to 200 minutes. That is like after 200 minutes, how many bacteria would be there? So, let us go back here. So, n at t equal to 200 minute is equal to 2 power, t is 200 minutes, 200 divided by 20 is 10, so, it's 2 power 10. So, there will be 2 power 10 bacteria. So, let us see how we can plot, when we plot this function n is equal to t into 2 power t by 20, what do we get? So, have a look at here, what we get is this kind of a function. So, this is what n is equal to 2n in the y axis and time in the x axis. So, after 200 minutes you have about more than 1000 bacteria. So, you can just calculate yourself and see what is 2 power 20, it has to be some number about 1000, where 1 over 20 is the rate of growth, 1 over 20 per minute, that is the rate of growth. So, now what we want to understand here, we want to understand something about the derivative, something about the slope of this curve. So, if you have a curve which, as you see here, which is exponential. So, let us just plot this curve here. So, we had time and number, and if we plot it, it is like exponentially growing curve. And if we calculate slope at every point, the slope at each point is different. The slope here is different, slope here, slope here. If you calculate the slope at each point, you can try calculating the slope, you will find that the slope itself is varying. So, if we want to get some idea about the slope, it is not very easy from this curve, because it is always difficult to calculate the slope of such a curve, like the slope at each place keeps varying. So, slope of this curve calculating is slightly difficult. So, people do a trick. So, the trick is the following. So, we can use this mathematical identity. So, what we know here is, we learn that the function we have is n is 2 power t by 20. Now, if you find log of this, if you find the logarithm log of n, so log n is log of 2 power t by 20. Now, it turns out that log of a power x is nothing but x log a. So, this is the rule of log of a power x is x log a. So, now using this rule here, log of t power 20. So, this is 20. So, let us say log of t power 20 is, so a here is 2, x is t power 20, this is our x. So, log of n is x log a, x is t by 20 into log 2. So, log 2 is a number, 20 is a number, t is the variable in the x axis. So, you can write it as actually t times log 2 divided by 20. So, basically log n is t times log 2 by 20. So, this is log n is t times some constant. Log 2 is a number, is 0.31, around 0.3 is log 2. Log, if you can look at what is log 2 in your calculator, I think it is 0.310 or some 3010 or something like that. So, it is about 0.3. So, log 2 is a number, 20 is a number. So, this is whole thing is a number. So, log n is some constant number times t. So, now let us try plotting. Instead of plotting n and t, let us plot log n and t. So, previously we had plotted n in the x axis and n in the y axis and t in the x axis, and we got a curve which is n and t, a curve which looked like this, and we had difficulty in calculating the slope, which is not very easy. One has to calculate slope at different points, and it is not the easiest way, easiest function to calculate the slope. Given the experimental data, calculating slope was little difficult. On the other hand, let us look here, let us plot log n versus t. So, let us plot these two things in the x, this in the x axis and this in the y axis. So, the expression we have is basically log n is t times log 2 divided by 20. This is the expression we finally got, and if we plot this as a y, this as x times some constant m, this is some constant. So, you know this, what is this? This is a straight line. What you would get is a straight line. So, let us see this plot. So, this is what, look at the plot in the left hand side, which is n is equal to 2 power t by 20, and in the in the right hand side you have log n versus time, and you get a straight line, and the slope of this straight line, the straight line is passing through 0, because there is no y y is equal to mx, is a line, straight line passing through 0, and the slope of this or the derivative of this is very easy to calculate, and you will see that the slope is log 2 by 20. So, if you know this log 2 by 20, immediately you can say that 20 is basically the rate of growth. So, from this measuring this, you can immediately calculate the rate of growth, which is 1 over 20. So, let us redo this a little more in general. So, in general, the expression for, so now for E. coli it is 20 per minute, 1 cell division in 20 minutes. So, it could be like for some other yeast it could be something else, for some other organism it could be some other number. So, the rate of growth in general could be any number k. So, it could be 1 per minute, it could be 2 per minute, it could be 10 per minute. So, let us call k as the rate of growth. So, which is something per second or something per minute, and we will have n is 2 power k t. So, this is the formula. We had k is 1 over 20. So, we had t by 20 before for E. coli. Now, if we take log on both sides, we have log n is equal to log of 2 power k t. So, log of a power x is x log a. So, this gives that log of n is nothing but k t times log 2. In other words, t times k log 2. So, the slope, so if you plot log n in the y axis and t in the x axis, the slope you will get is k log 2. So, slope you will get is k log 2. So, if you plot, so let us consider this. So, you have this equation which is y is equal to, so equation we have is log n is equal to t times k log 2. So, this is y is equal to x times m, where m is this quantity which is k log 2. So, as you can see, as we saw, if you plot this y and x, y equal to m x is a straight line, and the slope which is m, which is k log 2. So, now if you look at this here, if you look at this slide, what you had got is the slope as log 2 by 20. So, the slope we had got from this is log 2 by 20. So, instead of k log 2 here, the slope we got log 2 by 20. That means, k is 1 over 20. This means, k is 1 over 20. So, just by calculating the slope, so by plotting log n, so y is basically log n. So, y is equal to, y is nothing but log n. So, log n versus time, x is time. So, basically we plotted log n versus time. So, by plotting log n versus time, the log of the quantity, we got the slope by just calculating the slope of derivative of a straight line. Derivative of a straight line is the easiest thing to calculate. So, here again we were using the idea of, we wanted to calculate the slope, we want to calculate the derivative to get basically the rate of growth, and by doing this, what we did was, for doing to do this in the easiest way, we plot the log. So, this also tells you how to calculate the derivative of 2 power k t. So, now let us quickly again think about it. So, what we did is essentially calculating the derivative of 2 power k t, and by calculating the derivative we got the slope which is k log 2. So, now let us basically, this is another case where something in mathematics, some idea in mathematics is used. Basically, the derivative, we had some algebra plus some derivative finding slope is used to calculate certain rates in biology. So, here now, so with this we will stop the examples of derivatives for the moment, and we will go on to learn a few more other things today and go to the next topic in the next case. So, let us now look at a few another interesting cases of derivatives, positive derivative, negative derivative, zero derivative. So, let us think about slopes which are positive and negative and all that. So, let us take a curve. So, we know that straight lines have a constant slope. What about curves? So, let us look at this following slide. So, basically if you look at this curve and if you think about slopes at different places, what can we learn? So, let us in this curve, if you look at the slope here and if you look at the slope here and if you look at the slope here, what do we learn? So, let us quickly look at the slopes. So, the curve we have, so we have x axis and y axis, and we have a curve which look like this, and let us calculate the slope at different places. So, let us first calculate the slope here. So, let us calculate the slope here. So, this is the point which is that. So, now slope here is basically change in x, which is delta x, and change in y, which is delta y. So, basically we want to calculate delta x and delta y, and the slope is delta y by delta x. Without doing any calculation, we can say that since delta x is positive and delta y is positive, the slope has to be positive. So, this is some number which is positive number, and this is some number which is positive number. So, delta slope has to be positive. So, let us, in other words, let us do this. Delta x, when I say delta x is x 2 minus x 1. So, this is x 1, this is x 2, and this is y 1, this is y 2. So, the slope of the first case, so in the first case 1, slope is y 2 minus y 1 by x 2 minus x 1, which has to be greater than 0. If you do this, it is greater than 0, because as x 2 is larger than x 1 and y 2 is larger than y 1. So, as you can see here, x 2 is larger than x 1 and y 2 is larger than y 1. Now, let us look at the slope here. So, let us again redraw this curve for the convenience of calculating this, drawing this. So, let us plot x and y, and let us draw this curve, and if we calculate the slope here at this particular point, where the slope here. So, let us calculate the slope here. So, you have this two values, this is x 1 and this is x 2, but for each of this x 1 and x 2, the y value is same, the y does not change. So, y 1 equal to y 2. For x 1 you get the same y, for x 2 you get the same y. So, what does this mean? This means that y 1 and y 2 are same, that means y 2 minus y 1 is 0. So, the delta y is 0, that means slope is 0. So, it turns out that whenever they have a curve and at the maximum value of the curve, you have slope is 0. So, you had a positive, in this case you had positive slope. So, you had slope which is greater than 0. Here you have slope which is equal to 0. Now, let us look at the next case where we have delta x and delta y. So, let us again draw this. So, let us draw x and y and draw this curve, and let us look at the slope here. The slope here, so if we just mark x 1 and x 2, so let us mark this is x 1, the corresponding point is y1. So, this is y1. Now, let us mark x2. So, this is the value x2, the corresponding point here is y2. So, according to what we learned, the definition of derivative is dy by dx, which is what for whatever value of x1 we have y1. So, basically slope is y2 minus y1 divided by x2 minus x1. So, this is the derivative, this is the definition of slope. So, now let us look at what is x2 and what is y2. So, as you can see here, x2 is larger than x1. So, x2 minus x1 has to be positive. So, this is positive because x2 is larger than x1. On the other hand, y2 is smaller than y1. So, y2 minus y1 is negative because y2 is smaller than y1. So, if you this is a smaller number compared to this. So, if you subtract y2 y1 from y2 minus y1, this will be a negative number. So, the slope here, which is the slope here is negative, which is less than 0. So, the slope is less than 0 here. So, we saw 3 cases. So, let us look here. So, the definition, so this if you look at this thing here, here you saw that slope was positive. Here at the maximum, the slope was 0. That means, even if you change x, the y was not really changing. Here the x was increasing, y was also increasing. Here the x was increasing, but y was decreasing. So, the slope was negative. So, from a positive value, slope becomes 0 and then becomes negative. So, the slope decreases positive to 0 to negative, right. If you just plot the slope, and x, first it was some positive value, then it became 0, then it became negative. So, the slope was somehow decreasing. So, this kind of curves where the slope is decreasing is called convex shapes. So, you can see convex shape where the slope or the derivative is decreasing, and when the slope itself changing. So, as we all know, the slope is derivative. So, the derivative itself is keep changing. So, we can define something called curvature. So, let us look at this next slide. So, we have dy by dx, which is the slope. So, the dy by dx is changing from positive value to a negative value as we go along the x axis. So, as you can see that the slope itself is changing. So, what does it mean? The change in m, dm by dx. What does the dm by dx means? How does the slope changes? dy by dx is how does the y change? How does the y change is dy by dx, and dm by dx is how does the slope change? So, the slope changes is in mathematics, the slope change can be represented as d by dx of dy by dx, which is known as d square y by dx square. That is the change in slope, which is derivative of a derivative. So, m of x itself is a derivative. You again find the derivative of that, you will get d square y by dx square. That is called the curvature. So, we will discuss about this later, but just keep in mind for the moment, curvature is nothing but a second derivative of a derivative. That also has mathematical meaning. Mathematically, anything that is curved has a derivative of a derivative. Like you can calculate the second derivative and you will get either positive or negative. And what does it mean it is curved? So, the curvature has second derivative. We will come, and if we have time we will discuss how to calculate second derivative if by taking some examples. But for the moment let us just understand that there is something called second derivative, which is how does the slope itself change. So, the change in slope is d by dx of dy by dx, which is the second d square y by dx square. So, now you can imagine just the opposite. So, look at the next slide. What happens here? If you look at like what I did, if you just do all this, just plot such a curve, calculate the slope here, calculate the slope here and calculate the slope here, you will see that the slope is negative here, the slope is 0 here, the slope is positive here. So, the slope increases as you go along. So, the c of x, which is the second derivative, is positive and slope increases as we go along x. So, such things are concave shape. If you have a curve in concave shape, you will have slope that is increasing as we go along the x axis. So, just keep in, and another interesting thing which you should keep in mind is this derivative is 0 at the peak. Wherever you have a maximum or minimum. So, here you had a curve which is a maximum here and the derivative here was 0. In the next one you have a minimum here, the curve is changing from positive negative slope to positive slope and it has to cross through 0 somewhere, and the point of 0 crossing people call point of inflection, the slope is 0. So, two things you should understand from this, the last few slides of curvature is that the two take home message, two things you should change, you should understand is that whenever you have a curved function, a function that is not a straight line, any function that is not a straight line, the slope itself varies and sometimes the change in slope can be positive or negative. So, that you can calculate whether the change in slope is positive or negative by calculating the second derivative. Second derivative we will discuss how to calculate this clearly, but just keep in mind that we can calculate something called second derivative, which is d square y by dx square. That is, how does the first derivative, which is dy by dx, changes? How does the first derivative change? That is the second derivative, and the second derivative gives you the curvature. And at the point of whether it is maximum or minimum, whenever you have a curve has maximum or minimum, at that particular point the derivative is 0. So, this is three things which you should keep in mind. One is that the slope changes, as for any curve which is not a straight line, the slope will change and it can be positive change in slope or negative change in slope, and wherever there is a maximum the derivative is 0. We will use these things in the next coming lectures, So, for this moment, with this we will stop with this lecture here and we will go and learn more things in the coming lectures. Thank you.